What is aging?
Biological aging is not linear. We can move it back, we can move it forward, depending on many interventions. But we developed a small molecule in mice, and from the first time of administration, they live 73% longer. 73% is huge.
Irina Conboy is a world leader in the science of age reversal. As co-founder of Generation Lab, her groundbreaking research explores how the molecules in our blood and the noise they create could hold the key to staying young. Today, for the first time, she reveals a new technology that could extend our health span up to 73%.
Even today, when we bring this out, people still raise an eyebrow when we talked about it.
Can we identify the fundamental theory behind that and move aging back? The answer is yes. There are several clinics which are charging many thousands of dollars to become younger.
But is that true?
Scientifically, it makes sense. But there are side effects that become cumulative. Clinicians know those side effects. Our blood is a very dynamic system, but almost nobody paid attention to the more interesting point. The critical question is,
Hi, Dr. Irina. Welcome to the HumanWare Project recording.
thank you very much for having me and for giving me this opportunity to describe what I love to do, which is science.
According to you, what is aging?
Aging is a process, is a fundamental process that is closely linked to the second law of thermodynamics, or entropy. We become old simply because we eat and we breathe, and every time we do that in our mitochondria, energy factor of the cell, will produce molecules that damage the rest of the cell. And then that damage propagates. We also have external damage from UV light, right? UV rays. And so in general, we are regulated systems which can repair themselves and continue to be regulated, but not perfectly.
Right.
So with time, we fall apart. So that is my understanding of aging. Very interestingly, it is not just time-dependent process, because you can start with a young animal and make it prematurely old and senescent just within weeks. So there are options to control the process deliberately and slow it down.
When I first time to see that paper, what you do with Parabiosis, when you sew two mice together, I was very shocked. Could you tell us how that this study ever started?
So the work on heterochronic parabiosis actually started not just 20 years ago when it was published, but a few years before that. My husband and co-author on many of my papers, Dr. Michael Conboy, and I noticed that if you put brand new, healthy young cells in the environment when they have exposure to old blood, they become very quickly aged. So that immediately posed many questions, right? First of all, what is the temporal progression of aging if we can start with a young cell and make it old in days? How is this process controlled? Most importantly, will the same happen in vivo or in animal? Mike had this brilliant idea to connect young animal mice together in a procedure that is called Parabiosis, and then see if young mouse will become older and old mouse will become younger. So that's what precipitated this discovery. My whole involvement in the field of research was inspired by other scientists who did this before me. For example, Professor Cynthia Kinian from UCSF, she discovered that aging can be reversed in very small animals, worms, called C. elegans. And so that then inspired me to do research to see, can we do the same in mammals? Like humans are mammals and so are mice.
So when you start to have this question, do you sew those two mice by hands? I don't know, is that a stupid question?
It's not far-fetched. So what happened is that during the time that we had this question, there was another group of researchers, conveniently at Stanford, who were suturing two young mice together. The professor was on my PhD thesis committee, so I knew him very well. And therefore, I scheduled an appointment and I asked him, can you guys teach us how to suture the mice? We then connected many pairs of mice, and you're completely correct. You manually staple them. You staple. So you can imagine that you have two sleeping bags. You unzip them and then you zip them together. After 1 week, capillaries, small blood vessels, grow between the skin.
Yeah.
and then blood starts to be shed.
Yeah.
So it is not very well controlled procedure because you don't know when the positive effects start. And then you cannot do many other things, which then later on in the Conboy laboratory we improved what we can do. This work was groundbreaking in saying that you can start with an entirely old animal, and within a few weeks it will be younger. And that's what most people noticed. But almost nobody paid attention to the more interesting point, that you can start with an entirely young mouse, and within a couple of weeks it will become old.
But is that true? Even today, like when we bring this out, people still raise an eyebrow when we talked about experiment being successful every time, or is that too good to be true?
Awesome question, right? Experiment is successful every time. And to tell you the truth, there are very rare situations when experiment will be successful broadly and every time. Many people after our paper started doing the same work, connecting young and old mice, but studying different systems. For example, heart, or spinal cord, or gut, or skin. And in absolutely every situation, Biosis showed consistent rejuvenation of the old system. And again, nobody for some reason paid attention, but it showed premature aging of a young system.
Could you take us to a deep dive into those three organs and the parabiosis, what's going on?
Yeah, so the reason that we consistently study brain and liver is because they represent the three major layers of our tissues. So brain is ectodermal layer or external, outside. Muscle is mesodermal, meso in the middle.
Okay.
And then liver is endodermal or inside.
When you said those three layers, how does this being categorized? Is that because of the tissue or is that because of the blood?
There are many organs and tissues that we have, right? Mm-hmm. You have an option. It's a judgment call. There are many degenerative disorders of the brain that associate with aging. Then muscle is very important because old people have very low strength and they lose lots of muscle tissue, and that contributes to loss of their independence. And then liver is super important because, of course, aging of the liver is also well known. It is liver fibrosis and adiposity and metabolic decline. If we can rejuvenate those major organs, then chances are very high that the whole animal will be younger.
But how does that work for brain? Because when we talk about parabiosis, like the general idea, I do get the picture, but then when we say to really rejuvenate a certain organ, how does that work?
How does it work, right? Our hypothesis, even before the 2005 paper, was that aged blood is doing something horrible to young cells and tissues. And so then what happens is that if you connect young and old mice together, then young partner now is able to filter out all of those age-accumulated proteins from the parabiont.
Yeah.
And therefore making parabiont younger because systemic environment is calibrated or normalized to more youthful state. At the same time, young mouse suffers because it has to maintain old, inflammatory, afflicted body.
Yeah.
And now it has influx of all of these excessive proteins which inhibit regeneration of young tissues. The critical question is, do we even need young blood?
Logically, like if I'm getting older, I think I would need some younger or healthier, clearer blood in me to keep that field, like if I'm a car, right?
Yeah, exactly. So from kind of common sense logic, that's what people thought, and that's what Tony Weiss-Corey, Amy Wager, Sol Villeda decided to pursue. But scientific logic is very often different from common sense logic.
Okay.
It simply is based on a little bit better knowledge of those intricate molecules. And so scientifically, young blood and old blood are different not because you lose something with age, but because you actually have excessive amount of proteins. Too much.
Too much proteins.
Too much of certain proteins.
Okay.
Because of dysregulation of epigenetics and gene expression, you overproduce. And then at healthy young levels, each one of these proteins is indispensable for keeping you healthy. But when there are too many of them, then they become counterproductive.
Okay.
So from that perspective, adding even more to what you already have is not going to help you. You need to get rid of the excess and then calibrate system to how it was in your youth. Without any young blood, if you simply remove 50% in mice and 70% in people of blood plasma, which contains all of these proteins, then that is much more rejuvenative than adding young blood.
So could you take us into how did you turn your research from 20 years ago into a product that is actually changing people's health?
So once we figured out that in mice and in people, you can become younger simply through dilution of all plasma. You don't need anything young. That then poses the next question, can we simply keep diluting our plasma and become younger? By the way, the procedure is already FDA approved and was in practice, clinical practice, for over 30 years. Not to become younger, but to treat autoimmune diseases or blood diseases. But now, since 2020, there are several clinics which are charging people many thousands of dollars to perform the same procedure to become younger. I am not enthusiastic about it. I'm not really a big supporter. Because I think, again, scientifically it makes sense. But if you do a procedure once or twice in a very good clinic with qualified clinicians, it is relatively safe.
Right.
But if you do it round after round after round, then it becomes, basically there are side effects that become cumulative.
Right.
And the side effects are not very simple. For example, damage to liver and kidney, or toxicity from the blood thinners that are being added during this procedure. Clinicians know of those side effects. So I usually give these two metaphors. One is that if you have a fish tank, then you need to change water in the fish tank for fish to be alive and for plants to be alive. But if you can start changing it very frequently, the fish will die and the plants will die. And so basically plasma dilution, rejuvenation through plasma dilution, has this analogy, right? That we are changing water in a fish tank or we are changing oil in our car. Another analogy, which kind of goes back to your question, do we need young blood? Imagine that you have a cluster of rotten bananas and you try to make it better by throwing good banana in this cluster. Then, okay, it didn't work. Bananas are still rotten, right? And that's what all of these approaches are, right? So we have dysregulated system where we have excessive expression of genes, too many proteins are produced, they aggregate and cause all kind of neurodegenerative diseases. We try to solve this problem by adding something which is young.
Yeah.
It's not going to work. So that is the conundrum, right? It's a paradox. On one hand, we know the mechanism, what to do.
Yeah.
On the other hand, we don't have a method how to do it. We decided in the generation lab to say, okay, take advantage of our knowledge and develop the next generation longevity therapeutic, which is in process right now, and it's called GL1. GL stands for Generation Lab, and 1 stands for the very first product that we are going to have.
I do have some concern around it. If I exchange my blood plasma and then if I dilute it, will that causing any harm or side effect to it, or is it completely safe?
So with respect to simply diminishing the levels of proteins, it is not just safe, it is wonderful. Because they are diminished, so therefore they are no longer inhibiting abilities of tissue stem cells to repair tissues. And they also then cannot inhibit many other genes and proteins which go back to the same levels as when we were young. Because if you think about it, when we grow old, genes do not disappear from our blueprint. They are still in our DNA. They simply are not expressed because overproduced proteins block them. So once you remove this block, all of these young factors naturally go back. However, we don't have technology to keep doing it every month to keep a person in a young state. Many bad side effects will start to accumulate and invariably will overshadow any positive effect. And that's where GL1 comes into the play, right? This is why we are working on GL1.
Could you tell us what is GL1 and what does it do to us?
Yeah, so GL1 is a small molecule which by itself blocks the negative effects of age-elevated or excessive proteins on organs and tissues. And GL1 is proprietary to Generation Lab, but other molecules which are precursors of GL1 were tested in a Conboy lab in mice. And so mice are already old, they are analogous to about 75-year-old people and frail when we put them on GL1. And from the first administration, if you count from the first time of administration, they live 73% longer. 73% is huge because other potential longevity therapeutics usually produce about 15% maximum. And they're healthier. They have more endurance in treadmill, they have better short-term memory, they are less frail. So that is from the very first administration. But remember, this is mice, and there is still some caveats, and it doesn't work perfectly. So I don't want people to just say, okay, we need to grab whatever and start using for ourselves.
With your new product, is that a shot, like an aging shot, if I called it?
Yeah, so for some proteins, they will be still down at good levels for just a couple of weeks.
Yeah.
For others, maybe a month.
Okay.
Not very long. And if you want to normalize them broadly, then to be young, you need to do this procedure every month. But if you do it every month, you will be unhealthy. So then GL1 is a small molecule that is synergistic. It is working in the same direction as plasma dilution, but without plasma dilution. It is blocking many of the negative effects of accumulated proteins. And then when it is safe to do it again, maybe half a year from the first procedure, you can do it again and then inject GL1 again and again, and then continue getting gradually younger consistently.
Have you done the blood?
No, I'm waiting for clinical trials for GL1.
Okay.
Because of what I told you, right? I know that if I do it once, it will have certain positive effect, but it will not last.
Yeah.
And if I keep doing it many times, I will just start having health problems.
Do you think blood is the key to our vitality?
Yes, it is the key to our vitality and our health because it connects all of the organs and tissues. And if you have a problem in one part of your body, that will then propagate and cascade to another part of your body. And if you made your major organ like a liver healthier, that will systemically or through blood make other organs healthier.
When we think about blood, is the blood that goes through my body, they are always the same? Or when they go to different part of my organ, it will changing?
Yeah, it will change. So the systemic milieu or blood is very dynamic system, like most systems. Like even though you look at yourself in the mirror, you see the same face. But it's not the same face, it's not the same cellular responses, so everything in our bodies are constantly and dynamically changing. The whole system is stable because this dynamic is very, very precisely controlled, and we do not yet know how, and we do not yet have computer power to simulate it.
In Generation Lab, you do the biology age detecting and then to tell us about different organs age. Could you tell us what it is and how this is started behind it?
Approximately, 4 years ago, 5 years ago, people who are working on so-called DNA methylation clocks approached Conboy Lab and asked me to share our samples of all tissue, young tissue, and rejuvenated tissues so then they can tell me, based on their clock, the age of the tissue.
Okay.
And first I was happy about it. I thought, of course, guys, I will give you tissue, right? I will put cells or tissues in tubes and I will label them numerically and randomized. And then you will run them on your clock and tell me which one is young, right? Which one is old. And they're like, no, it's not going to work like that. You need to tell us which one is young, which one is old, and which one you try to rejuvenate. And that immediately had a huge question mark. If you have a way to predict biological age of a tissue, I don't have to tell you that, right? You already have a clock.
Yes.
So then we looked at what the clocks are, and what then turned out is that basically progression of biological aging is not linear. We know that young people are very different from old people in many, many parameters. But when we try to measure how do we go from being young to becoming old, we don't know. We know that it is not linear. For example, you don't get 1 gray hair per day. Clocks are linear models which are based on this fact that young is different from old. And then the rest of the data is mathematically manipulated to fit in between those 2 points. So they use this approach known as elastic net regression model, developed in 2005 by Hasty Group. And they apply it to the large data such as DNA methylation or proteins from people of different ages. And if you simply apply the data, it will not form the line. But if you start mathematically manipulating it, a filtration of the data, assigning some other weights, which has no biological justification, I basically realized that, okay, this is just a form fitting. What is much more interesting is what is the natural curve. I know that we can fit it, right, the data. But if you don't fit it, what is it? Hypothesis that we had and we published paper in 2023 is that it is not the levels of something. It is not like more or less of certain protein. It is dysregulation or noise, which biomarks biological age.
What is that noise?
Hypothesis is that, let's say that you have certain genes in your DNA blueprint that are critically important to be always at the same level. For example, they allow you to make energy or they control your heart health.
Right.
So if they are different level, people just die. So if certain genes exist, then whether person is 25 years old or 8 years old, the average level will be the same because they both are alive. But if you compare the deviation, standard deviation or dysregulation, then in the young people, they all will have kind of very, very small deviation and the level will be like that and they will all be within that range. And then all they will be like this. So there will be some people who have hugely different level. And that is what we call noise or dysregulation.
How long can we extend that mice life? Can we extend that?
Absolutely. Absolutely. In fact, DIL1 precursor that we have tested in mice, DIL1 precursor molecule extended lifespan and health span, not just how long they live, but how healthy they live, by 73% from the first administration of DIL1 precursor. What Generation Lab test is measuring is the level of entropy in your body. And not just in your body, but in the different systems of the body. If the whole entropy level is high, why? Is it because of the regeneration? Is now prematurely old and dysregulated. So there are 19 different functional systems, and we know the entropy of each one because we identified those critically vital pieces of our DNA, which on average are the same.
Right.
So dysregulation or noise is very, very easily to detect. We call them noise detectors.
Noise detector.
Yeah, so if you have something that is already noisy, you don't know, is it more noisy, less noisy? Every deviation from that stable line is easier to detect.
When we have different organs, maybe some organs they are aging slower, some organs are aging a lot faster than my current age. But if I know the metrics of each organ and I do some good intervention about it.
Yes, exactly. It's almost 100% accurate, right? Almost 100%. So what happens is that when we identify the natural pattern of aging, not fitting it into mathematical line, what turns out is that we start with a very low dysregulation when we are 25 to 27. Immediately at 31, it goes up. So when people typically drop out of Olympic teams, they cannot be top-class athletes like in gymnastics, they are less fit.
Yeah.
We don't notice that because to us they are extremely young, right? But they're already here. Then there is a healthy plateau when noise is pretty much stable, when our bodies are able to repair the entropy and to keep it good. Unfortunately, it is short-lived. Around 40, it goes up again to age-elevated disease risks. So age-elevated disease risk is increasing. And then there is another plateau of old age, which may or may not be the same for every population because our analyses are typically on relatively wealthy people who have luxury of maintaining good health. But this progression is surprisingly and remarkably the same as a pattern of aging when you look at the 460 areas of the genome, which is our proprietary noise detectors, or you split them into smaller groups, for example, tell you what's going on with your heart or your other systems of your body, immune system. And so what we can say, okay, based on all of them, you are on a healthy plateau, but based on one of them, you are already much, much noisier than your chronological age group.
There are the moments if somebody under a huge stress, their hair can go gray in very short period of time. What happened there? Is that because the noise is too much, so it can't maintain the same repair speed?
Yeah, you brought up this perfect example why aging is not linear. If you succumb to a disease, for example, COVID, you become older. If you recover from COVID, you become younger. And the same thing, the stress, right? So what happens is that, yes, progression of aging is not only non-linear, but it is dynamic, and we can move it back, we can move it forward, depending on many, many interventions. And Generation Lab has a list of really well-researched interventions for each deviation from healthy status. For example, there are supplements, omega, right? There are prescribed anti-inflammatory drugs that are already known. There are more intricate things which are medicine that clinicians know and can prescribe for patients. GL1 and GL2, GL3 and so forth will be the new class of the therapeutics that are based on the intricate understanding of how dysregulation happens during aging. And how, therefore, we can reduce the noise.
How did you see drugs like rapamycin?
I think that people should not take rapamycin.
Okay.
I don't think people should take metformin. The publications on rapamycin and metformin is the very interesting science in progress. But there was no reputable clinical trials which show success. If there are clinical trials, then I would be more relaxed about telling people that they could take those potential medicine. But rapamycin is known to inhibit immune responses and cause cancers. So it will depend on how do you use it. And then rapamycin inhibits signaling molecule called mTOR. But mTOR is not present at the same level in every cell. So the levels are constantly changing, and they need to change constantly for us to be healthy. Metformin inhibits the ability of mitochondria to make ATP. We need ATP. And if we don't have enough ATP, then tissue stem cells will not work to repair wounds. So even though there is amazingly interesting studies, which I think should be, fostered and moved forward to clinical trial, unless clinical trials are already published, I would not recommend people using those drugs.
When I think about our body being an anti-fragile system, it's supposed to rebalance back from whatever happened. And can we replace those juice? Like if I know what is stopping my body, is that like what's going
Unfortunately, first of all, what you are saying is totally true. And, a few years back when I started my scientific talks, I usually tell people when we are young, there is no pain, no gain.
Yeah.
And when we are old, there is lots of pain and still no gain, right? Unfortunately, it is not because of one thing. It would be great if you just say, okay, this is this one gene or protein which evolved to make us old or sick. Just remove it and then you become better. Unfortunately, there are hundreds of different changes. Can we identify the fundamental theory behind that and say, how can we meaningfully move aging back? The answer is yes, but it will not be by silver bullet.
How did you see science of anti-aging or aging will go in the future 5 years and our medical system?
So actually, let me start with saying that the first thing that I see is that there should be ton more funding for aging research. Because without that freedom to pursue your scientific ideas, people are pretty much kind of forced into dogma. For example, clocks are dogma. Well, think about it. Is it dogma or is it mathematical data fitting? And there are other ideas. For example, what is the natural pattern? Rapamycin or metformin or single bullet approaches is dogma. And if you had more funding, then we say, okay, we don't have dogma. You guys do your stuff, I do my stuff. We exchange information. And together we assemble this complex mosaic, which altogether tells us what should be done. So that is super important. For example, in 2023, we published that the key transitional points from being young to being old, we published them, right? It is 40 years old, is then the next transitional points between 50, 58, and the next point, right? Based on the noise increase. Then around 42, we go to higher noise. Then we have another plateau, and around 58, another very bad shift upward. So these same points that we discovered based on the natural pattern of biological aging DNA methylation were then a year later reported by Mike Snyder, right, at Stanford, with respect to clinical parameters. And if you think about it, this is awesome because both of our laboratories support each other finding, and we say this is really important, and you can discover it at the level of very, very first central dogma, very, very last downstream what happens to a person. However, these were completely different papers. They are not known in people's mind to represent the same story, right? And that is actually, that is a big problem. So Snyder Group, discussed our findings in their paper and said, by the way, this doesn't only happen with DNA methylation, it also happens with other important clinical parameters. It would have subtracted nothing from their glory, nothing. But now in this very challenging situation, perhaps an editor would say, oh, it's not novel then. So it is really kind of out of our hands.
Yeah.
And I'm fantasizing. I don't know what the editor would say.
What got you starting to study science at the very beginning stage?
So when I was like maybe 5 years old, back in the country which was called Soviet Union, I noticed that I'm getting taller. Like I could fit under the table, now I cannot fit under the table. I noticed that my grandmother has very wrinkly skin, and I kind of moved forward, hypothesized forward that the same will happen to me, then my grandmother will die. And from that point on, my motivation was always to understand how things work in biology and apply it to aging research. I enrolled in immunology program at Stanford during graduate school because the professors were the best, in immunology program, in my opinion. But even then, I continued discussions with my husband and with other scientists on how can we apply this and that to aging research.
If you will name one or two science paper related publishing that you have done in your career, what do you think is those heartwarmed or intelligent, intriguing paper that you have published?
So I think it will be 2003 science paper published in Science, where we show that if you just calibrate how cell to cell communicate, in old muscle becomes young. And that specific pathway was called notch. So that was the first idea that you don't need to exchange blood. You can just use pharmacology and you can rejuvenate tissue. But what inspires me, or what I think is the great discovery, might not be directly translatable to how I myself become younger, because I'm a scientist. So
Yeah.
for me, it's like, okay, this idea was proven, it is a great discovery. Others might think this is stupid discovery, or we cannot translate it to people.
Can I add on
Yeah.
to that? When you talk about that, it immediately flash back to my mind. I started learning about Chinese medicine with one doctor. They said that when you work out, your muscles start to getting very hard, and then when you touch a baby's skin, they are gooey, right? Like they are very soft. It's like fluid inside.
Baby skin is gooey because it has more protein called laminin and less protein called collagen.
Right.
And as we grow old, we have less and less of that flexible laminin, more and more of this tight collagen. And there are loads of things about Chinese medicine, for example, that are super interesting, very important to understand. It is just one muscle cell talking to another muscle cell.
Right.
And telling, I am injured, repair me. And then in aged muscle, if this is just one cell, this is another cell, they do not talk to each other. Right. And says, I'm injured, repair me, and this cell is doing nothing and sleeping. So then what we did, we then activated a protein, which now allows them to talk to each other, and then muscle was repaired better. So that was published back in 2003 in Science, and I think it was a big discovery, at least in my book. And then the second one was in 2022, which we published with Judy Campisi, who unfortunately passed away. She was, completely transcendental scientist. Her science goes on. And really invaluable mentor and a very good friend of mine. So we published this story in Nature Metabolism saying that if you have completely young, healthy mice and you exchange their blood, not suturing them, just blood exchange, with 50% of old blood, many organs and tissues such as kidney and liver and muscle become old within just a couple of weeks. So that was a big breakthrough because it kind of puts the nail on the coffin of young blood. Because they still have young blood, but they become old.
How old do you think we can live? In 50 years, how long humans' lifespan or health span can extend into?
I think it will be additional 40 years. So it will be, let's say, 120 years.
Okay.
But people will be healthy, they can play tennis, they can play golf. And it is not really far-fetched because just sanitation and nutrition extended our life from 40 where people were dying, to now, what is it, 80 if you can afford, right? So then it's another jump like that.
If you would give us one longevity or health aging related tips, what would that be?
So there is nothing better than healthy diet and exercise. Case in point, all of the scientists, famous scientists, are having healthy diet, sometimes caloric restriction, and exercise. And you would think that they know the whole field, right? So if there was anything else, they would do it. But, keep your eyes and ears open, and then hopefully GL1 and other Generation Lab products will come to market.
Thank you for sharing.
Thank you. Thank you for asking those great questions.
Yeah, thank you for your time today, Irina. Thank you for joining us for this episode of the Human Wear Project. Your subscription and a like means a lot to us, so that we can bring more content like this to you. Let's go on this journey together.